Hey guys, tonight we're going to float down into the midnight ocean, where the water is black as ink and cold as a forgotten thought, and watch jellyfish turn the darkness into something that looks like a constellation decided to go for a swim. You're suspended there in the deep, weightless, watching these translucent bells pulse through the water, trailing tentacles like silk ribbons, and suddenly, without warning, without sound, they ignite. Blue-green light ripples across their bodies in waves as if someone just flipped a switch inside a living lamp. It's beautiful, it's alien, and tonight, you're going to understand exactly why it happens. So, before you get comfortable, take a moment to like the video and subscribe, but only if you genuinely enjoy what I do here. And hey, drop a comment telling me where you're watching from and what time it is for you right now. I love seeing how far these bedtime stories travel while you're settling in for the night. Now, dim the lights, maybe turn on a fan for that soft background hum, and let's ease into tonight's journey together. You're drifting deeper now, and the pressure around you feels heavier, denser, like the ocean is gently reminding you that you're a visitor here. The jellyfish you're watching, let's call her Luna, because why not, is about the size of your palm, her bell contracting rhythmically as she propels herself through the water column. She looks fragile, almost impossibly delicate, like blown glass that might shatter if you touched it. But she's tougher than she appears. She's been around, in one form or another, for over 500 million years. That means her ancestors were glowing in ancient seas before there were fish, before there were eyes to see them glow, before the continents even bothered arranging themselves into anything you'd recognize on a map. Luna pulses again, and the light traces the edges of her bell like someone drawing with a neon pen in slow motion. You watch it spread, a ripple of cold fire that doesn't produce heat, doesn't consume fuel the way your campfire does, doesn't follow any of the rules you learned about light sources. It's bioluminescence, literally living light, and it's one of nature's most elegant party tricks. Here's what makes it genuinely strange. This light costs Luna almost nothing, while your phone's flashlight drains battery and gets warm, while your lamp turns electricity into heat and light at the same time, mostly heat if we're honest. Luna's glow is nearly 100% efficient. No wasted energy, no heat signature, just pure cold light, produced by a chemical reaction happening inside specialized cells scattered throughout her body. It's the kind of efficiency that makes human engineers weep quietly into their prototypes. You drift closer, careful not to disturb her, and you can see the individual photosites, the light-producing cells, dotted along the rim of her bell. They look like tiny pearls embedded in living jelly. When Luna wants to glow, and yes, she does want to, it's not some automatic reflex like your knee jerking when the doctor taps it with that little hammer. Though honestly, why do they still do that? These cells spring into action with remarkable coordination. The ocean around you is doing that thing where it shifts from black to deep purple to something that might be blue if you squint, and you realise you've been descending this whole time without noticing. You're maybe 200 metres down now, deep enough that sunlight is just a memory, a rumour of brightness that doesn't quite reach this far. This is where bioluminescence stops being a neat trick and starts being a survival strategy. Down here, making your own light isn't showing off, it's currency, it's language, it's the difference between eating and being eaten. Luna suddenly flashes brighter, a quick strobe that startles you even though you're just imagining this from your bed. Another jellyfish, let's call him Saul to keep the celestial theme going, has drifted too close. Luna's flash is a warning, or maybe a distraction, or possibly just her way of saying occupied, try the next current over. The light fades as quickly as it appeared, leaving ghost images floating across your vision, the way staring at a light bulb does before you remember that's a terrible idea. You watch Saul retreat, his own dim glow pulsing apologetically. Probably not, but you're projecting human emotions onto gelatinous invertebrates now, which means the sleepiness is setting in nicely. The interaction lasts maybe 3 seconds, but it tells you something important. These creatures aren't just passively glowing like nightlights someone forgot to turn off. They're actively using light to communicate, to defend themselves, to navigate a world where darkness is the default setting and vision is a luxury most creatures can't afford. Your eyes are adjusting now, the way they do when you wake up at 3 AM and stumble toward the bathroom without turning on the lights, and you start noticing that Luna isn't alone. There are dozens of them out here, maybe hundreds, each one a small lantern drifting in the current. Some glow steadily, others pulse in patterns that might be random, or might be coded messages you're not equipped to translate. A few stay dark, conserving energy, waiting for a reason to announce their presence to the void. The water moves around you in slow swirls, carrying plankton and debris and dissolved minerals and all the microscopic drama of ocean chemistry that makes this glowing possible in the first place. You're suspended in a living soup, which sounds less poetic than it should, but it's accurate. Everything down here is connected. The chemistry of the water, the biology of the jellyfish, the physics of light propagation through a dense medium. It's a system, intricate and ancient, that's been running smoothly since before your species figured out how to make fire look impressive. Luna drifts past your face now, close enough that you could count her tentacles if you wanted to commit to that level of meditative focus. You don't. Instead, you just watch the way her light reflects off the surrounding water, creating a small sphere of visibility in the otherwise impenetrable dark. It's peaceful down here, in a way that makes your bedroom feel cluttered by comparison. No notifications, no responsibilities. Just you and the glowing jellyfish and the gentle pressure of an entire ocean holding you in place. You're still floating there with Luna, and now you're wondering about the actual mechanics of this glow. Like, what's happening at the molecular level that turns a blob of transparent jelly into a living light bulb? Because it's not magic, despite looking exactly like magic, and the chemistry involved is weird enough to make you forget you're supposed to be falling asleep. Stay with me though. This gets good in a quietly nerdy way. Inside Luna's photosites, those pearl-like light cells you noticed earlier, there's a molecule called luciferin. The name comes from Lucifer, which means light bearer in Latin. And yes, someone with a decent sense of drama got to name this chemical. Luciferin is the fuel, the raw material that's about to get transformed. Sitting next to it, waiting like a patient catalyst, is an enzyme called luciferase. If luciferin is the log in your fireplace, luciferase is the match that sets the whole thing off, except this fire is cold, controlled, and happens to be occurring inside a gelatinous marine invertebrate drifting through the ocean at the speed of a gentle thought. When Luna decides it's time to glow, maybe because Saul got too close, or because something brushed against her tentacles, or because she's simply feeling conversational in the only language she knows, calcium ions flood into her photosites. This calcium surge is the trigger, the green light that tells Luciferase to get to work. The enzyme grabs the luciferin molecule and introduces it to oxygen, which in the chemical world is like introducing two people who are definitely going to hit it off, but in a very specific explosive way. What happens next is oxidation, which is just chemistry's word for burning without fire. The luciferin reacts with oxygen, catalyzed by that helpful luciferase enzyme, and in the process it transforms into oxyluciferin, luciferin's slightly more excited cousin. And here's where it gets beautiful. When luciferin becomes oxyluciferin, it has to release energy to make the transition work. In most chemical reactions, that energy becomes heat. Your metabolism burning glucose. Heat. Your car burning gasoline. Heat and noise and questionable emissions. But in bioluminescence, almost all that energy becomes light. Pure, visible photons radiating out into the water with virtually no thermal waste. You watch Luna pulse again, and now you're seeing it differently. Each flash is millions of these reactions happening simultaneously. A coordinated cascade of molecular events producing photons that travel at 792, 458 meters per second, the speed of light obviously, until they hit your retina, or a predator's eye, or the photoreceptors of some other creature trying to make sense of the darkness. The efficiency is genuinely absurd. Bioluminescence operates at about 90% efficiency, meaning 90% of the chemical energy becomes light, and only 10% becomes heat. Compare that to your standard incandescent bulb, which converts maybe 5% of its energy into light, and wastes the rest, making your lamp warm enough to remind you that entropy is real, and optimization is hard. Even your fancy LED bulbs only hit about 40% efficiency, and we invented those after landing on the moon. Well, decades after, but still. Luna's ancestors figured out 90% efficiency roughly 500 million years ago, presumably by accident, and then just kept it because evolution doesn't fix what isn't broken. You're drifting lower now, pulled by curiosity and the gentle current, and you notice that not all the glowing creatures down here produce the same colour light. Luna glows blue-green, that classic bioluminescent shade that science fiction movies love to use for anything alien or mysterious. But nearby, there's a jellyfish. We'll call her Stella because the naming convention we've established is clearly astronomical, who pulses in a slightly different hue, more blue than green, more sapphire than emerald, same basic chemistry, different specific molecules. Here's the thing about luciferin. It's not one molecule, it's a category, a whole family of light-producing chemicals that evolved independently across different species. Luna's luciferin is called curlanterrazine, which sounds like something you'd order at a fancy coffee shop, but is actually the most common luciferin in marine bioluminescence. Fireflies, which we'll visit later when we come back to the surface world, use a completely different luciferin. Bacteria use something else entirely. Evolution looked at the problem of how do we make light, and arrived at multiple solutions, like students in a chemistry class who all got the right answer using different methods and now refuse to acknowledge that the other approaches are equally valid. The colour of the light depends on the exact structure of the luciferin and the shape of the luciferase enzyme that's processing it. Change the enzyme's shape even slightly, and the photons come out with different wavelengths, different energies, different colours. It's like how a guitar string produces different notes depending on how tightly you've wound it. Same basic principle, different outputs. Luna's curlanterrazine produces blue-green light at around 470 nanometers, which happens to be the wavelength that travels farthest through seawater before getting absorbed. Convenient? Absolutely. Coincidence? Not even slightly. You watch another pulse of light roll across her bell, and you're starting to understand that what looks like a simple flash is actually a precision event, choreographed at the molecular level. The calcium surge, the enzyme activation, the oxidation reaction, the photon emission, it all happens in fractions of a second, controlled and repeatable, like a biological circuit board that runs on seawater and ancient genetic code instead of electricity and silicon. The water around you is full of dissolved oxygen, which is fortunate because bioluminescence requires it. No oxygen, no oxidation, no light. Luna's pulling oxygen from the surrounding seawater, processing it through her metabolism, and using it for both the boring task of staying alive and the more interesting task of glowing like a tiny supernatural chandelier. Multitasking at its finest, really. You think about your own body for a moment, the way your cells are also running oxidation reactions constantly, burning glucose with oxygen to produce the ATP that powers literally everything you do, including lying here imagining yourself as a deep-sea diver having revelations about jellyfish biochemistry. The difference is your oxidation produces heat and keeps you warm and occasionally makes you sweat during anxiety dreams about showing up to work without pants. Luna's oxidation produces light and makes her visible to anything within about 20 meters, which seems like a trade-off with distinct advantages and disadvantages depending on whether you're trying to hide or trying to be seen. Stella drifts past again, her slightly bluer glow trailing behind her like an afterthought, and you realise that you're watching chemistry happen in real time, watching molecules dance their specific dance and produce something as intangible and essential as light. It's alchemy, except it's real, except it's happening inside animals that don't have brains. Jellyfish are brainless, which honestly explains a lot about their life choices, except it's so elegant that human scientists spent decades trying to figure out how to replicate it in laboratories. You're floating closer to Luna now, close enough to see her body in detail, and you realise that understanding the chemistry is only half the story. The other half is anatomy, figuring out where exactly these light-producing reactions are happening, and how a creature without a brain manages to control them with such precision. Because Luna doesn't have a centralised nervous system, she doesn't have a control centre running the show, she's basically a sophisticated bag of water, with some specialised tissues thrown in, and yet she can turn her lights on and off like she's working a dimmer switch. Her body is simple in the way that elegant solutions are simple. The bell, that translucent dome you've been admiring, is mostly mesoglea, a jelly-like substance that's pretty much water held together by protein fibres and optimism. It's the biological equivalent of gelatin, except it's alive and functional, and doing a better job of staying intact than most gelatin you've attempted to unmold at family gatherings. The mesoglea gives Luna her shape, her bounce, her structure, but it's not where the light comes from. The photocytes, those specialised light-producing cells, are embedded in specific locations around her body, particularly along the rim of her bell. You can see them now, arranged in clusters like strings of microscopic Christmas lights. They're positioned strategically, not randomly, because even without a brain, evolution has a way of figuring out optimal placement through millions of years of trial and error and things that glowed in the wrong spots getting eaten before they could reproduce. When you look at the edge of Luna's bell, you notice a slight thickening, a ring of tissue that looks denser than the surrounding jelly. This is where most of her photocytes are concentrated. when she contracts her bell to swim, which she's doing now in that slow rhythmic pulse that's probably more relaxing than the meditation app you downloaded and used exactly twice. The mechanical pressure triggers those cells. Squeeze the bell, activate the photosites, produce light. It's a simple system, which is exactly what you want when you don't have a brain to micromanage everything. But Luna can also flash without swimming, which means there's another trigger mechanism at play. This is where her nerve net comes in. Not a brain, not even close, but a distributed network of neurons spread throughout her body like a democracy, where every cell gets a vote. When something touches her tentacles, those thread-like appendages trailing below her bell, the stimulus travels through the nerve net as an electrical signal, roughly the same way your own nerves work, but slower and less centralized, and probably less prone to anxiety about deadlines. The signal reaches the photocytes, calcium channels open, ions flood in, and suddenly Luna's glowing in response to being touched. It's a reflex, hardwired, automatic. Touch equals light. It's elegant because it doesn't require thinking, which is good, because thinking is not Luna's strong suit. She doesn't have a strong suit. She barely has suits at all. She's a jellyfish. You watch her tentacles drift past your face. You're still imagining this from your bed, remember, where it's safe and dry and significantly warmer than the actual deep ocean. And you can see that they're covered in tiny specialized cells called cnidocytes. These are the stinging cells, the ones that make jellyfish famous for ruining beach days and causing problems for swimmers who weren't paying attention. Each cnidocyte contains a coiled thread loaded with toxins, ready to fire when triggered. But right now, you're focused on how the tentacles also serve as sensor arrays, detecting pressure changes, chemical gradients, and physical contact in the dark water. When a small fish bumps into Luna's tentacles, several things happen almost simultaneously. The cnidocytes fire, injecting toxins. The nerve net activates, sending signals throughout her body. And the photocytes flash, creating a bright burst of light that serves multiple purposes. It might startle the fish, making it easier to capture. It might attract larger predators to the area, which could eat the fish and leave Luna alone. A strategy biologists call the burglar alarm effect, because apparently scientists enjoy mixing their metaphors across kingdoms. Or it might simply be a side effect of the activation cascade, light as a byproduct of being touched. You drift around to look at Luna from below, which feels vaguely intrusive, but she's a jellyfish and almost certainly doesn't care about your viewing angle. From this perspective, you can see her oral arms, the frilly structures surrounding her mouth, which also contain photocytes. The mouth itself is less of a mouth and more of a gastrovascular cavity opening, a hole that serves as both entrance and exit because Luna only needs one door in her architectural plan. Food goes in, waste comes out, same opening, zero complaints. It's efficient in a way that makes you question why vertebrates bothered with the whole separate entrance, separate exit thing. But evolution works with what it has, and Luna's ancestors apparently decided that simplicity was the way to go. The light from her oral arm serves a different purpose than the light from her bell rim. When she's captured something with her tentacles and is maneuvering it toward her mouth, the glow might help her navigate in the darkness, a built-in task light for eating dinner in the world's darkest restaurant. Or it might help attract more prey, because some small organisms are drawn to light like moths to flames, except wetter and with more immediate consequences. You notice that Luna's glow isn't uniform across her body. Some areas are brighter than others. Some pulse in patterns, while others stay steady. This variation comes from different densities of photocytes and different trigger thresholds in various parts of her nerve net. It's like how your phone's keyboard lights up when you touch it. Except Luna's been doing this since before keyboards existed, before phones existed, before the concept of communicating with light occurred to any creature with a spine. The bell contracts again, and you watch the light ripple from the center outward, a wave of bioluminescence that follows the muscle contractions. The mesoglea acts as a light pipe, channeling and diffusing the photons so they illuminate a larger area than just the photocytes themselves. It's fiber optics before fiber optics, biological light distribution that engineers studied when they were trying to figure out how to make their own systems more efficient. Nearby, a smaller jellyfish, let's call it Spark because we're running out of celestial names and also you're getting sleepy, produces a different pattern entirely. Sharp flashes, brief and intense, like a strobe light at a club where everyone is gelatinous and the music is just the ambient sound of water currents. Spark's photocytes must be wired differently. connected to a nerve net with different timing, different calcium dynamics, different evolutionary pressures that led to a different solution to the same basic problem of how do we use light to survive in the dark. You're realizing now that jellyfish anatomy, despite seeming simple, almost primitively simple, is actually exquisitely tuned for their environment. Every part serves a purpose. The bell shape maximizes swimming efficiency. The tentacle placement optimizes prey capture. The photosite distribution creates the exact light patterns needed for defense, hunting, and communication. Luna doesn't need a brain to coordinate all this because the system is self-organizing, distributed, resilient. It's the biological equivalent of a well-designed machine where every component does its job without needing a manager to oversee the process. You're still down here with Luna, watching her glow in the darkness, and now the obvious question floats into your mind like another bioluminescent creature drifting past. Why? Why go through all this effort? The specialized cells, the complex chemistry, the energy expenditure, just to produce light? Because evolution doesn't keep expensive traits around unless they're earning their keep, unless they're providing some survival advantage that outweighs the cost. And for Luna, glowing isn't just a neat party trick, it's a survival strategy that's been refined over hundreds of millions of years into something remarkably effective. Let's start with the most dramatic use: defense. You're floating in the dark ocean, and suddenly something large brushes against Luna's tentacles. Maybe it's a fish, curious or hungry, or just navigationally challenged. Luna's response is immediate. She flashes bright, a sudden burst of blue-green light that floods the surrounding water. The fish, expecting darkness and stealth and an easy meal, is suddenly confronted with what amounts to biological flash photography. It's startled, it's confused, and in that split second of hesitation, Luna contracts her bell and jets away into the darkness, putting distance between herself and the threat. This is called the startle response, and it works on the same principle as a car alarm going off when someone bumps your vehicle. Sudden noise, or in this case, sudden light, creates confusion and buys time for escape. The fish's eyes, adapted to the darkness, are temporarily overwhelmed by the brightness. By the time it recovers, Luna is gone, vanished into the black water like she was never there. It's not a guaranteed survival strategy, but it tips the odds in her favour, and in evolution, tipping the odds is all you need to pass your genes along to the next generation. But Luna has another defensive trick that's even more clever, and honestly a bit devious. When she's grabbed by a predator, let's say a larger fish that wasn't startled enough by the initial flash, she can produce a sustained glow. Not a brief flash, but a steady light that essentially turns her into a beacon. Now here's where it gets interesting. That light makes Luna visible. Yes, but it also makes the predator visible. Any larger predator in the area, something big enough to eat the fish that's eating Luna, can now see the lit up fish clearly against the dark background. It's like the fish is suddenly holding a neon sign that says "Fresh meal here, come and get it." This is the burglar alarm effect you heard about earlier, and it's genuinely brilliant in a dark evolutionary sort of way. Luna might get eaten, sure, but there's a decent chance the fish eating her will also get eaten, which means that fish's offspring won't inherit its Luna eating habits. Over thousands of generations, this creates pressure against eating glowing jellyfish, because the ones that glow when attacked are less likely to be eaten, and the predators that eat them are more likely to become prey themselves. It's mutually assured destruction, oceanic addition, and it works well enough that Luna's species is still here, still glowing, still deploying biological burglar alarms in the depths. Now let's talk about hunting, because Luna isn't just a passive drifter hoping food bumps into her tentacles, though that is part of her strategy, to be fair. Some jellyfish species use bioluminescence actively to attract prey. They pulse their lights in patterns that resemble the bioluminescence of plankton, or small prey organisms, essentially creating an underwater trap that says, "Hey, there's food over here," when really, they are the ones looking for food. Small fish and crustaceans, drawn to what they think is a meal, swim right into the tentacles and discover too late that they've made a significant tactical error. Luna herself doesn't hunt this way. She's more of an opportunistic feeder, grabbing whatever drifts into her tentacles. But you can see the strategy at work in other jellyfish nearby. There's one now, larger than Luna, with a bell the size of a dinner plate, pulsing in a slow, rhythmic pattern that looks almost hypnotic. Small organisms are drawn to it like moths to a porch light, except the porch light is carnivorous and has excellent capture mechanisms. It's lure fishing, biological addition. and it's been working for millions of years because prey animals apparently never got the memo about not trusting glowing things in the dark ocean. Communication is the third major use for bioluminescence, though calling it communication might be giving jellyfish too much credit for intentional signalling. Luna doesn't have complex thoughts about expressing herself through light, but her flashes do convey information to other creatures, including other jellyfish. When she flashes, she's essentially announcing her presence, marking her territory in three-dimensional space. Other jellyfish see this and adjust their swimming accordingly, avoiding collisions and spreading out across the water column to maximize feeding efficiency without competing for the same cubic meter of ocean. Some species seem to coordinate their flashing, creating patterns that ripple through groups of jellyfish like synchronized swimmers with bioluminescent costumes. Whether this is actual communication or just individuals responding to the same stimuli with similar timing is still debated by scientists who study these things professionally. Either way, the result is the same. The flashing helps maintain spacing between individuals and might even help them aggregate during mating season. Though jellyfish reproduction is weird enough that we should probably save that discussion for a night when you're not trying to fall asleep. You watch Luna pulse again, and you're starting to see her light as a language, or at least a signal system. Brief flashes mean back off. Sustained glow means help, I'm being eaten, come eat my attacker. Rhythmic pulsing means this space is occupied, try elsewhere. It's not as sophisticated as human language, obviously. Luna can't glow in Morse code to spell out the ocean is particularly cold tonight, or I missed the Cambrian period when everything was simpler. But within the constraints of her nervous system and her ecological niche, she's using light to survive, and that's all evolution asks. There's also the possibility, and this is more speculative, that some bioluminescence is essentially accidental, a side effect of other metabolic processes that happen to be useful enough to stick around. Not every flash needs a purpose. Not every glow needs an explanation rooted in survival advantage. Sometimes chemistry just produces light, and if that light doesn't actively hurt the organism's survival chances, it persists through the generations because evolution is more concerned with removing harmful traits than optimizing every single feature to perfection. But mostly, when you watch Luna and her glowing neighbours, you're watching organisms that have turned light into a tool, a weapon, a lure, a shield, a signal. They're manipulating photons the way you manipulate words, using them to interact with their environment in ways that increase their chances of surviving long enough to reproduce. And they're doing it without brains, without conscious thought, without ever taking a biology class or reading a paper on optimal defense strategies. They're just glowing. Because their ancestors who glowed survived better than their ancestors who didn't, and that's the only logic evolution needs. You're drifting upward now, rising slowly through the water column like a thought surfacing from deep sleep, and you're leaving Luna behind in her dark world. But before you surface completely, before you leave the ocean entirely, there's one jellyfish you need to meet, one specific species that changed everything. Not just for jellyfish, but for human medicine, for biology, for the way scientists understand living cells. You're heading toward the coast of the Pacific Northwest, toward waters that are colder and shallower, where a particular jellyfish has been quietly glowing for millions of years, waiting for someone to notice that its light was more than just beautiful. The jellyfish is called Equaria victoria, and if you passed it on a beach, you probably wouldn't give it a second glance. It's translucent, unassuming, about the size of your fist when fully grown, with a flattened bell and delicate tentacles that trail behind it like afterthoughts. It doesn't look revolutionary. It looks like every other small jellyfish bobbing in the shallows, the kind that swimmers mostly ignore unless they're worried about stings, which Equaria victoria barely delivers. Its sting is so mild that most people don't even notice it, making it possibly the most polite jellyfish in existence. But here's what makes it special. Equaria victoria glows green. Not blue-green like Luna down in the depths, but a bright, vibrant green that looks almost artificial, like someone painted it with highlighter ink. And this green glow, this specific shade produced by this specific jellyfish, contains a protein that would eventually win scientists a Nobel Prize and revolutionize the entire field of cell biology. You're looking at one of the most important jellyfish in scientific history, and it doesn't even know it, because again, no brain. The story starts in the 1960s with a scientist named Osamu Shimomura, who was working at Princeton and had become fascinated with bioluminescence. He wanted to understand the chemistry behind it, to isolate the molecules responsible for producing light. So he did what any dedicated researcher would do. He went to Friday Harbour, a marine laboratory in Washington state, and started collecting Equoria victoria by the thousands. And when I say thousands, I mean he and his team collected tens of thousands of jellyfish over several summers, squeezing them to extract the bioluminescent material, working with buckets and nets and a determination that borders on obsessive. What Shimamura found was colenterrazine, that same luciferin Luna uses down in the deep ocean, producing the initial blue light through the chemical reaction you learned about earlier. But Equoria victoria had something extra, something unexpected. The blue light from the colenterrazine was being absorbed by another protein and re-emitted as green light. This secondary protein was acting as a light converter, taking blue photons and spitting out green ones, and Shimamura had no idea what it was or why it existed. He isolated the protein, purified it, and discovered that it glowed green even without the bioluminescent reaction. Just shine blue or ultraviolet light on it, and it fluoresced, absorbed the light and re-emitted it at a longer wavelength. This was different from bioluminescence, which produces light through chemical reactions. This was fluorescence, which is more like a molecular echo, light bouncing back at you in a different colour. Shimamura called it green fluorescent protein, or GFP, because scientists are practical people who name things descriptively and save the creative flourishes for their research papers. For years, GFP was just an interesting curiosity, a weird protein from a jellyfish that glowed green for reasons nobody fully understood. Shimamura published his findings, the scientific community nodded politely, and everyone moved on to other projects because a glowing protein seemed neat but not particularly useful. And then, in the 1990s, a researcher named Douglas Prasher figured out how to clone the gene that codes for GFP, meaning he could now insert this gene into other organisms and make them produce the protein. Suddenly GFP became the molecular biology equivalent of a miracle. You could tag genes with GFP, attach it to specific proteins, and watch them glow inside living cells. You could see where proteins travelled, how they interacted when they were turned on or off. Before GFP, studying proteins inside living cells was like trying to watch a movie in a dark theatre while blindfolded. You knew something was happening, but you couldn't see the details. GFP turned on the lights. It made the invisible visible. Martin Chalfie demonstrated that GFP could work in other organisms by making bacteria and roundworms glow green. Roger Tsien, a chemist, figured out how to modify GFP to create different colours – blue, yellow, cyan, giving scientists a whole palette of fluorescent proteins to work with. Together, Shimamura, Chalfie and Tsien won the Nobel Prize in Chemistry in 2008 for their work on GFP, which by that point had become one of the most widely used tools in biological research. You're floating next to an Equoria victoria now, watching it pulse through the shallow water, its green glow visible even in the dim light filtering down from the surface. This jellyfish, this specific species, has been photographed more times than most celebrities. It's appeared in thousands of scientific papers. Its genetic code has been inserted into mice, fish, monkeys, plants, bacteria, and probably a few organisms that scientists aren't publicly discussing, because the ethics committees would have questions. There are mice out there right now that glow green under UV light because they have jellyfish genes in their DNA. There are fish whose neurons light up when they fire, letting researchers watch thoughts happen in real time. There are plants that glow when they detect pollutants, turning environmental monitoring into a light show. All of this traces back to Equoria victoria, to Shimamura spending his summers squeezing jellyfish in Washington state, to a green protein that evolution created for reasons that still aren't entirely clear. Why does Equoria victoria have GFP? The most likely explanation is that it converts the blue bioluminescent light into green, because green travels better through the coastal waters where it lives. Blue light gets scattered and absorbed quickly in shallow water full of sediment and algae. Green light penetrates farther, lasts longer, makes the jellyfish more visible for whatever purpose its bioluminescence serves. Probably the same defense and communication functions Luna uses in the deep ocean, just optimized for a different environment. But here's the beautiful part. Evolution created GFP for Equoria victoria's purposes, and humans borrowed it for entirely different purposes, purposes that evolution never anticipated and couldn't have planned for. The jellyfish is glowing to survive. Scientists are using that glow to understand cancer, to track neural connections, to visualize how embryos develop, to watch viruses infect cells in real time. It's tool repurposing on a molecular scale, taking something nature built for one job and discovering it's perfect for another. You watch the jellyfish pulse again, its green glow washing over you like a gentle reminder that sometimes the most important discoveries come from the most unlikely places. Not from dramatic deep-sea expeditions or high-tech laboratories, but from buckets full of jellyfish collected at a marine station. From a scientist curious enough to ask, why does this glow? And patient enough to spend years finding the answer. You're still near the surface, the water brighter here, warmer, and you're thinking about that green fluorescent protein, GFP, sitting inside Equirria victoria, waiting to be discovered. But now you need to see what happened after the discovery, after scientists figured out they could borrow this jellyfish protein and insert it into practically any living thing. Because this is where the story shifts from interesting marine biology fact to technology that fundamentally changed how we understand life. And honestly, it's one of those rare moments where the hyperbole is actually justified. Picture a laboratory, anywhere in the world, sometime in the late 1990s. A researcher is trying to understand how a specific protein moves through a cell. Before GFP, this was almost impossible to track in living tissue. You could kill the cells, stain them with dyes, slice them thin, examine them under a microscope. But that only showed you frozen moments, snapshots of a dynamic process. It was like trying to understand a dance by looking at still photographs. You could guess at the movement, infer the pattern, but you weren't really seeing it happen. Now with GFP, the researcher can fuse the gene for the target protein to the gene for GFP. When the cell produces the protein, it also produces the green fluorescent tag attached to it. Turn on a blue or UV light, and suddenly you can watch the protein move through the living cell in real time. It glows, it traces paths through the cytoplasm, it clusters at the cell membrane or migrates to the nucleus, or does whatever proteins do, and for the first time, you're actually watching it happen instead of guessing based on dead cells and statistical analysis. This is what scientists call a revolution, though they usually use more technical language and publish it in journals with names like Nature and Cell, where excitement is expressed through carefully chosen adjectives like significant and unprecedented. But make no mistake, being able to see inside living cells changed everything. It changed how we study disease, how we develop drugs, how we understand basic biology that's been happening inside organisms for billions of years, but was invisible to us until a jellyfish protein made it glow green. You're watching light filter through the water now, and you're imagining those early experiments, bacteria engineered to glow green, just to prove it could be done. The bacteria didn't care. They went about their bacterial business, dividing and metabolizing and doing whatever bacteria do, except now they glowed under the right light. Then came the roundworms, those tiny transparent nematodes that researchers love because you can see through their bodies. Insert GFP into specific cells, just the neurons say, and suddenly you have worms whose nervous systems glow green, letting you watch neural circuits function in a living, moving animal. Then came the mice. Researchers created mice that glowed green in every cell. Mice whose cancer cells glowed so doctors could track tumour spread in real time. Mice whose immune cells lit up when they encountered infections. These weren't Frankenstein experiments, despite what the more excitable corners of the internet suggested. These were tools, ways of seeing processes that were always happening but were previously invisible. The mice weren't harmed by the glowing. GFP is biologically inert, just sitting there fluorescing without interfering with normal cellular processes, like the world's most useful passenger. And then scientists got creative, because that's what scientists do when you give them a useful tool. Roger Tien, remember him from the Nobel Prize trio, started tweaking the GFP molecule, changing amino acids here and there to see what would happen. Some changes made it brighter, some made it dimmer, and some changed the colour entirely. He created cyan fluorescent protein, yellow fluorescent protein, and eventually dozens of variants spanning the spectrum from blue to red. Now researchers could tag multiple proteins in different colours and watch them interact like a molecular light show. Picture a neuron now. One of your neurons maybe, except it's been engineered for research purposes so parts of it glow different colours. The cell body glows green. The axon, the long cable that sends signals to other neurons, glows red. The synapses where it connects to other neurons glow yellow. Under the right microscope you can watch signals travel down the axon in real time, watch the synapse light up when neurotransmitters are released, watch the whole communication network of the brain doing its job while it's still alive and functioning. This is how we're mapping neural circuits now, how we're understanding brain function, how we're tracking what goes wrong in diseases like Alzheimer's and Parkinson's. Cancer research exploded with new possibilities. Researchers could tag cancer cells with one colour and immune cells with another, then watch the battle happen in living tissue. They could see which cancer cells were dividing, which were dying, which were metastasizing to other parts of the body. Drug companies could test new treatments and watch in real time whether the drugs reached their targets, whether they killed cancer cells or just made them glow angry colours without actually helping. Developmental biologists, the people who study how a single fertilised egg becomes a complete organism, suddenly had the ability to watch embryos develop with different tissues glowing different colours. Heart cells in red, brain cells in green, muscle cells in blue, all forming and organising and somehow knowing where to go and what to become. It's like watching time-lapse footage of a building constructing itself, except the building is alive and the construction crew is made of cells following genetic instructions that have been refined over millions of years. Environmental scientists got in on it too. They created bacteria that glow when they detect specific pollutants: arsenic, mercury, other contaminants that shouldn't be in drinking water. Drop the bacteria into a water sample, and if it glows, you know there's a problem. It's faster than traditional chemical testing and sensitive enough to catch contamination at very low levels. Farmers started using plants engineered with fluorescent proteins that change colour when the plant is stressed. Not enough water, nutrient deficiency, disease. The plant essentially tells you what's wrong by changing which colour it glows, like a biological diagnostic system that requires zero medical training to interpret. And here's what makes this especially beautiful. None of this required inventing new physics or chemistry. Scientists didn't have to create fluorescent proteins from scratch. They just borrowed something that already existed, something that evolved naturally in a jellyfish bobbing around the Pacific Northwest coast, minding its own business and glowing green for its own jellyfish reasons. Evolution did the hard work of creating a stable, bright, non-toxic fluorescent protein. Humans just had to notice it, understand it, and figure out how to use it for purposes that have nothing to do with jellyfish survival. You're floating on your back now, looking up through the water at the surface, at the wavering light that breaks and shifts with each ripple. You're thinking about Shimmer squeezing thousands of jellyfish, probably wondering if this tedious work would lead anywhere interesting, probably not imagining that decades later his discovery would be helping doctors track cancer metastasis, and engineers design biosensors, and teachers show students what cellular machinery looks like when it's actually working. The jellyfish next to you pulses again, its green glow soft and steady, completely unaware that its evolutionary adaptation has become humanity's window into the microscopic world. It's just glowing because that's what it does, because its ancestors who glowed survived better than those who didn't, because evolution built this tool and humans found a use for it that spans from basic research to clinical medicine to environmental monitoring. You're ready to move on now, to leave Acheria Victoria and its revolutionary green glow behind, because the ocean is full of other creatures that light up the darkness in ways that are equally beautiful and strange. You're drifting through coastal waters now, through zones where sunlight still penetrates but weakly, where the line between photosynthesis and bioluminescence starts to blur, and you're about to meet jellyfish's cousins, creatures that look similar, that occupy similar ecological niches, but that aren't technically jellyfish at all, even though most people call them that anyway because taxonomy is complicated and common names are forgiving. First up are the comb jellies, or ctenophores, which have been causing taxonomic headaches for scientists since someone first looked at them under a microscope and said, "Wait, these aren't jellyfish. They look like jellyfish, translucent, gelatinous, drifting through the water with an otherworldly grace, but they're in a completely different phylum, as distinct from true jellyfish as you are from a sea urchin." Evolution, it turns out, really likes the transparent blob with tentacles body plan and has invented it multiple times independently, which should probably tell us something about optimal design for slow-moving marine predators, but mostly just makes biological classification annoying. You're watching one now, let's call it Prism because of what you're about to see. Prism is about the size of a walnut, oval-shaped, with 8 rows of cilia running down its body from top to bottom, like meridian lines on a globe. These cilia are its locomotion system, tiny hair-like structures that beat in coordinated waves to propel Prism through the water. And here's the magical part. When light hits those beating cilia, they diffract it, splitting white light into rainbows that ripple down Prism's body in waves of colour. red, orange, yellow, green, blue, violet, cycling through the spectrum like a living prism, which is why you named it that. This isn't bioluminescence, technically. Prism isn't producing light, it's just bending it, manipulating it through the physics of diffraction. But the effect is so striking that many people assume it's some form of biological glow. In bright light, near the surface, prism shimmers with iridescent bands of colour that pulse as the cilia beat. It's pure physics. The spacing of the cilia is just right to split visible light into its component wavelengths. But it looks like magic. Like Prism is wearing a coat made of moving rainbows. But wait, because Prism also has actual bioluminescence. When threatened, say when something bumps into it, or when you reach out to touch it in your imagination, it produces a blue-green glow similar to true jellyfish. The photocytes run along its 8 comb rows, and when activated they create lines of light that trace Prism's body like glowing racing stripes. So Prism has 2 light shows, the rainbow diffraction in bright water, and the bioluminescent glow in darkness. It's frankly showing off at this point, using both physics and chemistry to manipulate light in ways that make it visible and memorable, and hopefully less likely to be eaten. Nearby you spot something larger, a ribbon of translucent tissue undulating through the water like a silk scarf caught in a breeze. This is a siphonophore, and it's about to complicate your understanding of what counts as a single organism. Because this ribbon, this creature you're looking at, isn't one animal, it's a colony. It's dozens, maybe hundreds of individual polyps, called zooids, that have specialised into different roles and fused together to create what looks like a single organism, but is actually a cooperative of tiny animals working together. Some zooids handle swimming, contracting rhythmically to propel the colony forward. Others handle feeding, trailing tentacles that capture prey. Still others handle reproduction, budding off new zooids to grow the colony. And scattered throughout are zooids specialised for bioluminescence, producing light when the colony is disturbed. It's like if your body was made up of hundreds of tiny people, each one doing a specific job, all of them so interdependent that they can't survive separately. Which honestly isn't that far from how your own cells work, if you think about it long enough. You're also a colony, just a colony of cells instead of a colony of polyps, all of them cooperating to create the illusion of a unified individual. The siphonophore pulses with light now, a wave of blue luminescence rippling down its length in response to a current shift. Each glowing zooid activates slightly after its neighbour, creating a travelling wave of light that makes the whole colony look like a neon sign flickering on one bulb at a time. The effect is mesmerising, hypnotic even, and you have to remind yourself that you're supposed to be getting sleepy, not more awake with fascination at colonial invertebrates and their light displays. You drift deeper again, following the siphonophore as it descends, and you realise that some of these colonies can grow to enormous lengths. The longest siphonophore ever recorded was over 40 metres, longer than a blue whale, longer than most buildings are tall, just spiralling through the deep ocean like a living light rope, trailing tentacles and glowing when disturbed. It's one organism colony thing, depending on how you define organism, and it's one of the longest animals on Earth, and most people have never heard of it because it lives where humans rarely visit, and would fall apart if you tried to bring it to the surface. There are other glowing gelatinous creatures down here too, salps, which are actually more closely related to you than to jellyfish, because they have a notochord at some point in their life cycle, making them chordates, distant cousins sharing an ancient ancestor. Salps form chains, linking together in colonies that can stretch for meters, each individual pumping water through its body to filter feed and to jet propel the chain forward. When threatened, the chain lights up, individual salps glowing in sequence or all at once, depending on the species and the nature of the threat. You watch a salp chain pulse past, each individual about the size of your thumb, their bodies so transparent you can see their internal organs as dark shapes suspended in clear jelly. The bioluminescence, when it activates, comes from bacteria living inside them, symbiotic partners that produce light in exchange for nutrients and a safe place to live. It's a partnership, a deal worked out over evolutionary time, where both parties benefit enough that neither has evolved the ability to survive without the other anymore. The water around you is full of these drifting, glowing, gelatinous things. Not all jellyfish, not all even closely related to each other, but all converging on similar solutions to similar problems. How do you catch food when you're slow and don't have teeth? Be transparent, drift with the current, extend tentacles, wait for something to swim into them? How do you avoid being eaten when you're soft and defenseless? Glow when attacked, startle predators, attract their predators, make yourself visible in ways that create problems for anything trying to eat you. It's a whole ecosystem of glowing jellies, each species filling a slightly different niche, each one using light in slightly different ways for slightly different purposes. Prism diffracts sunlight and glows blue when touched. The siphonophore colony coordinates its bioluminescence across hundreds of zooids to create travelling waves of light. The salp chain relies on bacterial partners to produce its glow. And true jellyfish like Luna and Equoria victoria use their own photosites with their own chemical reactions to create light on demand. You're rising toward the surface now, breaking through into air, leaving the ocean behind for a moment because bioluminescence isn't just a marine phenomenon. Evolution discovered how to make light multiple times, in multiple kingdoms of life, in environments that have nothing to do with water or pressure or the deep dark sea. You're standing on land now, let's say it's a warm summer evening, the kind where the heat of the day lingers in the air and crickets are beginning their nightly chorus, and you're waiting for darkness to fall completely so you can see what else glows when the sun goes down. There, in the grass near your feet, a small light blinks on. Then another. Fireflies, or lightning bugs if you grew up calling them that, rising from the vegetation like tiny handheld stars that someone released into the air. They're beetles, not flies. Taxonomic names are misleading like that, and they're using bioluminescence for a completely different purpose than jellyfish use it. Luna glows to defend herself, to startle predators, to survive. These fireflies glow to find mates, to advertise themselves, to say, "Here I am, and I'm the species you're looking for, and if you're interested in reproduction, flashback." You watch one drift past at eye level, its abdomen glowing yellow-green with that soft pulsing rhythm that makes summer evenings feel magical. The light organ is on its underside, a specialized structure filled with photosites that work on the same basic principle as jellyfish photosites. Luciferin plus luciferase plus oxygen equals light. Except firefly, luciferin is chemically different from jellyfish colinterazine. Evolution invented bioluminescence separately in insects, came up with different molecules to accomplish the same goal, because apparently there are multiple solutions to the problem of how do we make living organisms glow. The firefly, let's call him Flicker, because we've committed to this naming convention and there's no backing out now, is flashing in a specific pattern, on for half a second, off for 2 seconds, on again, off again, maintaining this rhythm as he flies in a swooping path above the grass. He's calling out in light code, broadcasting his species identity and his availability and his general coordinates to any female fireflies who might be watching from the ground. Different firefly species have different flash patterns, different durations, different intervals, different colours even, all to make sure they attract the right mates and don't waste time courting fireflies they're incompatible with. Somewhere in the grass below, a female firefly is watching Flicker's display. She's staying put, conserving energy, waiting to see if his pattern matches her species code. If it does, she'll flash back, usually a different pattern, a response that says, "Yes, I see you, I'm interested, come closer." If Flicker sees her answering flash, he'll adjust his flight path, home in on her, location, and if everything goes well, they'll mate and produce the next generation of bioluminescent beetles who will glow in the same patterns, because those patterns are coded in their genes like dance steps passed down through generations. Except sometimes things don't go well. There are predatory fireflies, femme fatales of the insect world, who have learned to mimic the flash patterns of other species. A male firefly, thinking he's found a receptive female of his own species, follows the flashes to the ground and discovers too late that he's been lured by a predator who promptly eats him. It's dark, it's efficient, and it's a reminder that bioluminescence can be exploited, can be faked, can become a weapon instead of a communication tool. Evolution is full of these twists, these arms races where one species develops a trait and another species figures out how to manipulate that trait for its own benefit. You're walking now, through the summer evening, watching fireflies rise around you like reverse falling stars. Their light is remarkably efficient. About 95% of the energy in the chemical reaction becomes light, with only 5% wasted as heat. This makes fireflies even more efficient than Luna the jellyfish, even more efficient than your best LED technology, and they're accomplishing this with organic chemistry happening inside beetle abdomens. If human engineers could replicate this efficiency in commercial lighting, we'd save enormous amounts of energy. But despite decades of research, we're still not quite there, still learning from beetles how to make cold light that doesn't waste power heating the surroundings. Now you're leaving the open field, walking into forest, and the light changes. Not firefly light this time, but something dimmer, more diffuse, glowing from the ground itself. You kneel down, letting your eyes adjust, and you see them. Mushrooms, small and nondescript in daylight, now glowing with a faint green luminescence that makes them look like they've been painted with weak, phosphorescent paint. These are bioluminescent fungi, and they're using light for reasons that scientists are still debating, because fungi don't have mates to attract and don't have predators to startle, and generally don't seem to benefit obviously from glowing in the dark. The leading theory is that the light attracts insects, which then spread the fungal spores to new locations, helping the fungus reproduce. The mushrooms you're looking at are the fruiting bodies, the reproductive structures, and making them glow might help them disperse their spores more effectively than non-glowing mushrooms. It's a hypothesis, not proven, but it makes sense in an evolutionary way. Traits persist when they provide some advantage, and if glowing mushrooms spread their genes more effectively than dark mushrooms, then glowing becomes common in the population. You touch one gently, and your finger comes away with a faint smudge of glow, the bioluminescent chemicals transferring to your skin briefly before fading. Fungal bioluminescence uses yet another type of luciferin, completely different from firefly luciferin and jellyfish calenterazine. Because evolution keeps reinventing this wheel, keeps finding new molecular pathways to the same destination of produce light without heat. There are over 80 species of bioluminescent fungi known to science, scattered across different continents, growing on decaying wood or emerging from forest floors. Some glow continuously throughout the night, others pulse or flicker in response to disturbances. A few glow brightly enough to read by, though you'd need a lot of them, and probably very good eyesight and questionable reading priorities to attempt this. Most just produce a faint glow that's easy to miss unless you're looking for it, unless you're paying attention to the small, strange lights at your feet. In some parts of the world, Japan, Brazil, parts of Southeast Asia, bioluminescent mushrooms are common enough that locals have known about them for centuries, have names for them in their languages, have folklore explaining why the forest floor glows in certain seasons. In other regions, they're rare enough that most people have never seen them, have no idea that fungi can produce light at all, would probably assume you were making things up if you mentioned glowing, mushrooms in casual conversation. You stand up, brush the dirt from your knees, and keep walking. The forest around you is dark now, fully night, and you're aware of all the bioluminescence you can't see. The bacteria glowing in rotting wood, too faint for human eyes to detect. The insect larvae glowing to attract prey or deter predators, hiding in leaf litter where you'd never notice them. The millipedes and centipedes and other small arthropods that produce bioluminescence for reasons that are probably very important to them but completely mysterious to scientists who study these things. On land, bioluminescence is rarer than in the ocean, but it's still there, still happening, still being used by creatures who found ways to make light in environments where light is scarce. The fireflies are still flashing in the field behind you, their yellow-green pulses continuing through the night until dawn, or until they find mates, whichever comes first. The mushrooms will keep glowing, spreading their spores through visiting insects, doing their slow fungal work of decomposition and reproduction, and you'll keep walking, watching for small lights in the darkness, understanding now that bioluminescence isn't confined to the ocean, isn't owned by jellyfish, but is a solution that life discovered multiple times, in multiple forms, wherever there was an advantage to producing light without heat. If human engineers could replicate this efficiency in commercial lighting, we'd save enormous amounts of energy. But despite decades of research, we're still not quite there, still learning from beetles how to make cold light that doesn't waste power heating the surroundings. Now you're leaving the open field, walking into forest, and the light changes. Not firefly light this time, but something dimmer, more diffuse, glowing from the ground itself. You kneel down, letting your eyes adjust, and you see them. Mushrooms, small and nondescript in daylight, now glowing with a faint green luminescence that makes them look like they've been painted with weak, phosphorescent paint. These are bioluminescent fungi, and they're using light for reasons that scientists are still debating, because fungi don't have mates to attract and don't have predators to startle, and generally don't seem to benefit obviously from glowing in the dark. The leading theory is that the light attracts insects, which then spread the fungal spores to new locations, helping the fungus reproduce. The mushrooms you're looking at are the fruiting bodies, the reproductive structures, and making them glow might help them disperse their spores more effectively than non-glowing mushrooms. It's a hypothesis, not proven, but it makes sense in an evolutionary way. Traits persist when they provide some advantage, and if glowing mushrooms spread their genes more effectively than dark mushrooms, then glowing becomes common in the population. You touch one gently, and your finger comes away with a faint smudge of glow, the bioluminescent chemicals transferring to your skin briefly before fading. Fungal bioluminescence uses yet another type of luciferin, completely different from firefly luciferin and jellyfish calentorazine. Because evolution keeps reinventing this wheel, keeps finding new molecular pathways to the same destination of produce light without heat. There are over 80 species of bioluminescent fungi known to science, scattered across different continents, growing on decaying wood or emerging from forest floors. Some glow continuously throughout the night, others pulse or flicker in response to disturbances. A few glow brightly enough to read by, though you'd need a lot of them, and probably very good eyesight and questionable reading priorities to attempt this. Most just produce a faint glow that's easy to miss unless you're looking for it, unless you're paying attention to the small, strange lights at your feet. In some parts of the world, Japan, Brazil, parts of Southeast Asia, bioluminescent mushrooms are common enough that locals have known about them for centuries, have names for them in their languages, have folklore explaining why the forest floor glows in certain seasons. In other regions, they're rare enough that most people have never seen them, have no idea that fungi can produce light at all, would probably assume you were making things up if you mentioned glowing mushrooms in casual conversation. You stand up, brush the dirt from your knees, and keep walking. The forest around you is dark now, fully night, and you're aware of all the bioluminescence you can't see. The bacteria glowing in rotting wood, too faint for human eyes to detect. The insect larvae glowing to attract prey or deter predators, hiding in leaf litter where you'd never notice them. The millipedes and centipedes and other small arthropods that produce bioluminescence for reasons that are probably very important to them but completely mysterious to scientists who study these things. On land, bioluminescence is rarer than in the ocean, but it's still there, still happening, still being used by creatures who found ways to make light in environments where light is scarce. The fireflies are still flashing in the field behind you, their yellow-green pulses continuing through the night until dawn, or until they find mates, whichever comes first. The mushrooms will keep glowing, spreading their spores through visiting insects, doing their slow fungal work of decomposition and reproduction, and you'll keep walking, watching for small lights in the darkness, understanding now that bioluminescence isn't confined to the ocean, isn't owned by jellyfish, but is a solution that life discovered multiple times, in multiple forms, wherever there was an advantage to producing light without heat. You're descending again, back into the ocean, but this time you're going deeper than before, much deeper. You're dropping past the zones where sunlight penetrates, past the twilight realm where Luna lives, past the continental shelf and into the true abyss where the pressure is crushing, and the temperature hovers just above freezing and the darkness is absolute. This is the deep sea, and down here, bioluminescence isn't occasional or decorative. It's everywhere. It's standard. An estimated 90% of the organisms in the deep ocean produce light in some form, which means you're entering the largest bioluminescent habitat on Earth, a realm where darkness is punctuated by countless tiny lights, each one a creature trying to survive in an environment that seems fundamentally hostile to life. You're sinking past 200 meters now, past 500, past 1000, and the water around you is black in a way that surface darkness never is. There's no ambient light, no reflected glow from the surface, no moon or stars filtering through from above. This is pure absence of light, the kind of darkness that would drive surface-dwelling creatures insane, and yet down here, life is abundant. It's just using light differently, producing its own illumination for reasons that are often stranger and more creative than anything Luna or the fireflies came up with. Something drifts past your face. A jellyfish. Yes, but not like any you've seen before. This one is deep red, almost maroon, and it glows with a red bioluminescence that looks like dying embers. Most bioluminescence in the ocean is blue or blue-green because those wavelengths travel farthest through water. But red light gets absorbed quickly, doesn't travel far, which makes it almost invisible to most deep-sea creatures whose eyes are tuned to detect blue light. This jellyfish, call it crimson, has essentially discovered stealth lighting, a way to illuminate its surroundings that only it can see, like having night vision goggles in a world where everyone else is looking for blue lights. Crimson uses its red glow to hunt. It shines red light on potential prey, illuminating them without them knowing they're being lit up, because they can't see red wavelengths. It's like using infrared to hunt in the dark, except the infrared is actually visible red light that only appears invisible because evolution didn't give most deep-sea creatures red light receptors in their eyes. Crimson can see them. They can't see the light, and by the time they realise something's wrong, they're already caught in Crimson's tentacles. It's clever, it's predatory, and it shows that bioluminescence isn't always about being visible. Sometimes it's about selective visibility, about being seen only by the organisms you want to see you. You keep descending, and now you spot something that looks like it came from a nightmare designed by someone who understands too much about deep-sea adaptation. It's an anglerfish, female, about the length of your forearm, with a mouth full of teeth that point inward. Teeth designed to grip prey that tries to escape. Teeth that say, if you get caught, you're staying caught. But the remarkable part is the lure dangling in front of her face, hanging from a modified spine that extends from her forehead like a fishing rod. The tip of the lure glows blue-green, pulsing gently, mimicking the movement and light signature of small prey organisms. Smaller fish, drawn to what they think is food, or maybe just curious about the light in the darkness, swim closer to investigate. The anglerfish stays perfectly still, conserving energy, letting the lure do all the work. When prey gets close enough, and we're talking centimetres here, almost touching distance, the anglerfish strikes with remarkable speed, jaw extending, teeth closing, prey disappearing into a mouth that seems disproportionately large for the fish's body. The whole encounter takes maybe a second. The light keeps glowing, the lure keeps waving, and the anglerfish waits for the next curious victim. You're still deep, still surrounded by darkness punctuated by strategic lights, and now you're noticing something you've seen hints of already, but haven't fully explored. Many of these glowing creatures aren't making light themselves. They're farming it. They're hosting it. They've entered into partnerships with bacteria that produce bioluminescence, and these partnerships are so intimate, so essential, that neither partner can really survive without the other anymore. You're watching symbiosis in action, evolution's way of outsourcing light production to specialists who do it better than their hosts ever could. Remember that anglerfish with the glowing lure? The light came from bacteria. The fish provides a specialised organ, essentially a tiny house with blood supply and nutrients, and the bacteria move in and produce light in exchange for room and board. It's like having a tenant who pays rent by keeping the lights on, except the tenant is billions of microscopic organisms and the rent is bioluminescence. The bacteria in the anglerfish's lure belong to a genus called Photobacterium, which is exactly the kind of straightforward naming that happens when scientists discover something and want everyone to know immediately what it does. These bacteria are free living in the ocean as well, floating in the water column, producing light that seems wasteful when they are alone but makes perfect sense when they're in large enough numbers. See, individual bacteria glowing in isolation doesn't accomplish much. The light's too faint, too scattered to be useful. But when bacteria reach a certain population density, when enough of them are clustered together, their combined glow becomes visible, becomes functional, becomes something that other organisms notice. This population-dependent glowing is called quorum sensing, which sounds like a democratic process for bacteria to vote on whether to turn the lights on, and that's not entirely inaccurate. The bacteria produce and release signalling molecules as they multiply. When the concentration of these molecules reaches a threshold, meaning when enough bacteria are present, the genes for bioluminescence activate simultaneously across the population. Suddenly, millions of bacteria glow together, creating light that's bright enough to matter, bright enough to be seen, bright enough to attract the attention of fish and squid and other potential hosts who might provide better living conditions than drifting alone in the cold, dark water. You're watching this happen now in a small squid called a bobtail squid. Not a deep-sea species, actually, but one that lives in shallow waters and comes out at night to hunt. The squid is about the size of your thumb, translucent enough that you can see its internal structures, including a specialised light organ in its body cavity that glows with blue-green light. Inside that organ are Vibrio fischeri bacteria, a different species than the anglerfish bacteria, but operating on the same basic principle. The squid provides nutrients and a home, and the bacteria provide bioluminescence that the squid uses for counter-illumination. The partnership starts fresh with each generation. Baby bobtail squid hatch without bacteria. Their light organs are empty, sterile, waiting. The squid release chemical signals that attract free-living Vibrio fishery from the surrounding water. The bacteria swim toward the signals, enter the light organ through special pores, and colonize the space. Once they reach quorum, once enough bacteria are present, they begin glowing, and the squid gains its bioluminescent capability. Every morning, the squid expels most of the bacterial population, keeping just a small seed colony. Every night, that colony multiplies back to full strength and resumes glowing. It's a daily cycle of partnership renewal, population control, and mutual benefit. Why do the bacteria agree to this arrangement? Because inside the light organ, conditions are ideal. There's a steady supply of nutrients. The squid's immune system doesn't attack them because they're recognized as beneficial partners. The environment is temperature controlled and safe from predators that would happily eat free-floating bacteria. From the bacteria's perspective, living inside a squid's light organ is prime real estate, worth the cost of producing bioluminescence in exchange for security and resources. And why do squid bother maintaining bacteria instead of evolving their own bioluminescence? Because outsourcing is efficient. Evolving and maintaining the genetic machinery for bioluminescence takes resources, takes evolutionary time, takes energy that could be spent on other survival-related traits. By partnering with bacteria who already do this well, the squid gets the benefits of bioluminescence without the costs of producing it from scratch. It's specialization, division of labor, exactly the kind of efficiency that makes symbiotic relationships so common in nature. You drift past other creatures using the same strategy. There are flashlight fish, named for the obvious reason that they have glowing patches under their eyes that look like built-in flashlights. The glow comes from bacteria living in specialized organs, and the fish can control the light by either expelling some bacteria to dim it, or by covering the light organ with a dark membrane, like closing a shutter. They use the light to hunt at night, to communicate with each other, to navigate in the darkness. Bacteria just glow, doing their bacterial thing, unaware that they've become part of a fish's sensory equipment. Some pinecone fish have light organs in their jaws filled with bioluminescent bacteria. When they open their mouths, the light shines out, illuminating prey or possibly confusing predators who weren't expecting a fish's mouth to double as a flashlight. Certain squid species have multiple light organs scattered across their bodies, each one colonized by bacteria, each one serving a different function. Some for counter-illumination, some for startling predators, some for species recognition and social signaling. The partnership between hosts and bioluminescent bacteria is so successful that it's evolved independently multiple times across different groups of organisms. Fish discovered it, squid discovered it, even some invertebrates like tunicates host bioluminescent bacteria. Each partnership is slightly different bacterial species, different light organs, different mechanisms of control. But the core principle remains the same. Bacteria produce light, hosts provide resources, both parties benefit enough that the relationship persists across generations and becomes hardwired into the biology of both partners. And here's what's particularly fascinating. The bacteria pass down the genes for bioluminescence vertically through their lineages, while hosts acquire bacteria horizontally from the environment. This means that the bacterial species have been glowing for millions of years, evolving their light-producing capabilities long before any fish or squid thought to partner with them. The bacteria didn't evolve bioluminescence to serve hosts. They evolved it for their own reasons, possibly to promote DNA repair, possibly as a byproduct of metabolism, possibly for functions we still don't fully understand. Hosts just recognize something useful and built partnerships around it. You're surrounded now by creatures lit from within by bacterial tenants, and you're realizing that bioluminescence in the ocean is often a collaborative effort. It's not just individual organisms producing light through their own chemistry, but entire ecosystems of partnership, symbiosis, mutual dependence where the boundaries between host and symbiont blur into something more integrated, more interdependent than simple landlord-tenant relationships would suggest. You're floating upward now, beginning the long ascent back toward the surface, back toward the world of sunlight and air and human civilization. But before you leave the ocean entirely, before you forget about Luna and Crimson, and all the glowing creatures you've met in the depths, you need to understand what happened after scientists learned about bioluminescence, after they isolated GFP and figured out how these light-producing systems work. Because this knowledge didn't stay confined to marine biology textbooks and academic journals. It escaped the lab, jumped disciplines, and ended up changing medicine in ways that would have seemed like science fiction a few decades ago. You're in a hospital now, or rather you're imagining one, because we're pivoting from jellyfish to human applications, and somehow you're managing this transition smoothly despite being half asleep. A doctor is preparing for surgery, but this isn't ordinary surgery, this is cancer removal, and the surgeon needs to see exactly where the tumour is, needs to distinguish cancerous tissue from healthy tissue with precision that matters at the cellular level. Here's where bioluminescence enters the picture, specifically GFP and its fluorescent cousins that scientists have been engineering and improving since Shimmer first isolated the protein from Equoria victoria. The patient received an injection days ago, a dose of genetically modified viruses carrying the gene for a fluorescent protein. These viruses are designed to target cancer cells specifically, to infect tumours but leave healthy tissue alone. Once inside the cancer cells, the viruses insert their genetic payload, and the cancer cells start producing fluorescent proteins. They glow, not with their own bioluminescence, human cells don't have luciferin or luciferase, but with fluorescence lighting up when the surgeon shines blue or ultraviolet light on them during the operation. The surgeon can now see the tumour margins clearly, can identify even tiny clusters of cancer cells that would be invisible to the naked eye, can remove malignant tissue while preserving as much healthy tissue as possible. It's precision surgery guided by borrowed jellyfish technology, and it works remarkably well for certain types of cancer, brain tumours especially, where distinguishing tumour from healthy brain tissue is critical, and mistakes have serious consequences. This isn't science fiction, this is happening now, in hospitals around the world, using fluorescent proteins derived from jellyfish and coral and other marine organisms that glow naturally. The proteins have been modified, optimized, made brighter and more stable, but the core technology comes from creatures that never intended their glow to help humans perform surgery. Evolution created these proteins for one purpose, and humans repurposed them for another, and somehow it works better than anyone initially expected. Beyond surgery, fluorescent proteins are revolutionizing drug development. Pharmaceutical companies can tag specific proteins in disease pathways with GFP, then test thousands of potential drugs to see which ones affect those proteins. If a drug successfully blocks a disease protein, the glow changes. Maybe it gets brighter, maybe dimmer, maybe it relocates to a different part of the cell. Scientists can watch this happen in real time, can screen drugs faster than ever before, can identify promising candidates without killing cells and staining them and examining them under microscopes in processes that used to take weeks but now take hours. You're in a research lab now, watching a mouse run through a maze. The mouse looks normal under regular light, but when you switch to ultraviolet illumination, its brain lights up with multiple colours. Different neurons glow different hues, some green, some yellow, some cyan, each one tagged with a different fluorescent protein. Researchers are watching the mouse think, watching neural circuits activate as it makes decisions, watching memory form in real time as cells light up in patterns that correspond to learning. You're still drifting through the deep, and now you're starting to wonder about something that you've noticed but haven't questioned yet. Why is most bioluminescence blue? Luna glowed blue-green. The anglerfish's lure glowed blue-green. The bacteria in the squid produced blue-green light. Even the fireflies back on land glowed yellow-green, which is close enough to be in the same general region of the visible spectrum. There's physics behind this, rules about light and water and wavelengths that explain why evolution keeps arriving at blue as the default colour for underwater bioluminescence. Light is just electromagnetic radiation, photons vibrating at different frequencies, and the frequency determines the colour you see. Red light has a longer wavelength, about 700 nanometers, which means the photons are spread out, oscillating more slowly. Blue light has a shorter wavelength, around 475 nanometers, with photons packed closer together, vibrating faster. When light travels through water, different wavelengths behave differently. Water molecules absorb and scatter light, and they're particularly good at absorbing the longer wavelengths like red and orange. Red light dies quickly in water, usually within the first few meters of depth. Blue light, though. Blue light travels farther, penetrates deeper, maintains its intensity better than any other visible wavelength. This is why the ocean looks blue when you're snorkeling in clear water. You're seeing the blue wavelengths that haven't been absorbed yet, the light that's still bouncing around looking for something to illuminate. And this is why bioluminescence evolved predominantly in the blue-green range, particularly in the ocean. If you're a jellyfish trying to communicate through bioluminescence, producing red light would be pointless because it wouldn't travel far enough for anyone to see it. Producing blue light, though, that signal carries farther, reaches more potential receivers, gets absorbed less by the surrounding medium. Evolution optimizes for efficiency, and in the ocean, blue is the most efficient colour for light-based communication. You watch Luna pulse again, she's still here with you somehow, or maybe you've cycled back to her depth zone, and her blue-green glow spreads through the water in a sphere maybe 10 meters in radius before fading to invisibility. That's actually impressive penetration for biological light. Compare it to red light, which would barely make it 2 meters before being completely absorbed, and you start to understand why crimson, that deep red jellyfish you met earlier, evolved red bioluminescence specifically as stealth lighting. Red doesn't travel far, which means crimson can see her surroundings without announcing her presence to every predator in the neighbourhood. The colour of bioluminescence also depends on the specific chemistry involved. Remember how luciferin and luciferase react to produce light? The exact structure of those molecules determines what wavelength of light gets emitted. Change a few amino acids in the luciferase enzyme, alter its shape slightly, and suddenly the reaction produces photons with different energies, different wavelengths, different colours. This is how fireflies produce yellow-green light instead of blue. Their luciferin and luciferase are structured differently than marine versions, tuned to produce longer wavelengths that stand out better in air than in water. Some organisms have evolved multiple versions of luciferase, allowing them to produce different colours depending on which version they activate. There are jellyfish that glow blue when startled, but can switch to green for sustained illumination. The blue flash is for immediate defence, the startle response that might save them from an attack. The green glow is for the burglar alarm effect, sustained visibility that attracts secondary predators. 2 colours, 2 functions, both optimise for survival in different threat scenarios. In air, the physics are different. Light doesn't get absorbed as quickly, because air molecules are less dense than water molecules, less likely to intercept photons and convert them to heat. This means fireflies have more flexibility in their colour choices. Yellow-green stands out well against dark foliage, is easily visible to other fireflies, and requires slightly less energy to produce than blue light would. Some firefly species produce orange or even reddish glows, though these are rarer because they're harder for firefly eyes to detect at distance. You're breaking the surface now, emerging into air, and you're shifting focus one more time because bioluminescence isn't just helping humans in laboratories and hospitals, it's also helping us understand the ocean itself, the planet's health, the changes happening on scales too large for any single person to observe directly. Scientists have discovered that bioluminescent organisms can serve as indicators, as living sensors that reveal environmental conditions, climate patterns, and ecosystem health through their presence, their behaviour, their glow. You're floating on your back now, looking up at stars that are just beginning to appear as twilight deepens. Below you, in the water, dinoflagellates are starting their nightly performance. These are single-celled organisms, neither plants nor animals, but something in their own category, and they produce bioluminescence when disturbed by movement, waves, boat wakes, swimming fish, your hand trailing through the water. Each disturbance triggers a flash, a burst of blue-green light that lasts a second or two before fading. When conditions are right, when nutrients are abundant and water temperature is optimal, dinoflagellates bloom in enormous numbers, creating bioluminescent bays where every wave glows, every splash creates ripples of light, every movement in the water becomes visible through the sparkle it generates. You've probably seen videos of this. Bioluminescent bays in Puerto Rico, beaches in California where the surf glows blue, dolphins swimming through water that lights up around them like they're trailing stardust. It's beautiful, genuinely magical looking, and it's also telling scientists important information about ocean conditions. Dinoflagellate blooms occur when specific conditions are met: the right temperature range, sufficient nutrients, calm enough waters for the cells to accumulate. When these blooms happen in unexpected places or at unexpected times, they signal changes in ocean chemistry, water temperature shifts, nutrient flows altered by currents or runoff or climate patterns. Some dinoflagellate species produce toxins during blooms, the infamous red tides that can poison shellfish and harm marine life. The bioluminescent displays often accompany these toxic blooms, serving as a visible warning system that something unusual is happening in the water. Fishermen and coastal communities have learned to watch for the glow, to avoid harvesting seafood during bioluminescent bloom events, to treat the light show as both beautiful and cautionary. It's environmental monitoring through bioluminescence, nature providing its own early warning system for ecological disruption. Scientists are using bioluminescent organisms as bioindicators for pollution as well. Some species of bacteria only glow when they're healthy, when environmental conditions fall within their optimal range. Expose them to heavy metals, pesticides or other pollutants, and the glow dims or stops entirely. Researchers have created biosensor systems using these bacteria, essentially living test strips that measure water quality by reporting how brightly they glow. Drop the sensors into a water sample, wait for the bacteria to respond, and the light intensity tells you whether the water is clean or contaminated. It's faster than chemical analysis, cheaper than sending samples to laboratories, and it works in remote locations where sophisticated equipment isn't available. Bioluminescent plankton distributions are also revealing ocean current patterns and climate-related changes. Certain species thrive in specific temperature ranges and water conditions, so tracking where bioluminescence appears and disappears helps scientists map how water masses are moving, how temperatures are shifting, how climate change is affecting ocean stratification. Satellites can detect large-scale bioluminescent displays from space, the combined glow of billions of organisms bright enough to register on sensors orbiting hundreds of kilometers above Earth. These satellite observations are being integrated into climate models, helping researchers understand how warming waters affect plankton distributions, which in turn affect the entire marine food web since plankton form the foundation of ocean ecosystems. There are also creatures that produce multiple colours simultaneously by layering different light-producing systems. Some siphonophores have blue bioluminescent zooids, but also contain green fluorescent proteins similar to Equoria victoria's GFP. The blue light from bioluminescence excites the fluorescent proteins, which re-emit the energy as green light. The result is a creature that glows in both blue and green, creating a two-tone light display that's distinctive enough to serve as a species identifier, useful when you're a colonial organism trying to find other colonies of your own species in the dark ocean for potential reproduction. Then there's the truly unusual case of the stoplight loosejaw fish, which you're looking at right now because one just swam past your position. This fish has red bioluminescent light organs under its eyes, extremely rare for deep-sea organisms, and here's the clever part. It also has visual pigments in its eyes that can detect red light, which most deep-sea fish lack. This gives the loosejaw a private communication channel, a wavelength that can see but its prey can't. It's like having infrared goggles in a world where everyone else is colorblind to infrared. The loosejaw shines red light on potential prey, sees them clearly illuminated, and strikes before they even know they've been spotted. The stoplight loosejaw gets its red bioluminescence through a different pathway than most organisms. Instead of producing red light directly through chemical reactions, it has a filter system. The light organs initially produce blue-green light, the standard bioluminescent color, but then that light passes through a filter containing red pigments that absorb the blue wavelengths and let only red pass through. It's biological color correction, like putting a red gel over a blue flashlight, and it's one of the few examples of red bioluminescence in the deep ocean that isn't produced directly by chemistry. You're starting to see that color in bioluminescence isn't random or aesthetic. It's functional, optimized by evolution for specific purposes in specific environments. Blue dominates in the ocean because blue travels farthest through water. Red appears occasionally as stealth lighting for predators who've evolved the ability to see it. Green shows up through fluorescent protein intermediates that modify blue light. Yellow-green appears in fireflies, and some shallow water organisms where air or less dense water allows for more color flexibility. Even the intensity matters. Bright flashes for defense, attracting attention. Startling predators. Dim glows for counter-illumination, matching background light, becoming invisible through camouflage. Pulsing patterns for communication, each species with its own rhythm, its own signal that says this is what I am, this is where I am. The deep sea is full of light signals, each one carefully tuned to propagate through seawater at the right wavelength, at the right intensity, for the right purpose. You close your eyes for a moment. You're getting sleepy now. The information saturation reaching that pleasant point where your brain is full of interesting facts, but also ready to process them during sleep, and when you open them again, The bioluminescence around you seems less random, more organised. Each light is a message, each colour a choice, each wavelength optimised by millions of years of trial and error and survival pressure to be exactly what it needs to be for the organism producing it. This is neuroscience in the 21st century, and it's only possible because jellyfish evolved green fluorescent protein for reasons that have nothing to do with helping humans understand consciousness. The technique is called brainbow, which is exactly the kind of whimsical name you get when scientists who are tired of technical terminology get to name their own project. By expressing multiple fluorescent proteins in different combinations, researchers can colour-code individual neurons, making each one glow a unique hue. This lets them trace neural connections through the brain, map circuits with precision that was impossible before fluorescent proteins became available. They're building atlases of brain connectivity, understanding how neurons wire together to produce behaviour, thought, memory, emotion. Diagnostic medicine is changing too. There are biosensors now, living cells engineered with fluorescent proteins that change colour in response to specific molecules. Want to detect glucose levels in diabetic patients? Create cells that glow brighter when glucose is present, dimmer when it's absent. Want to identify bacterial infections quickly? Engineer fluorescent proteins that activate only in the presence of specific bacterial toxins. The cells become living diagnostic tools, reporting on chemical environments by changing their glow, translating molecular events into visual signals that doctors can interpret without complex lab equipment. Some hospitals are testing fluorescent protein-based sensors that can be implanted under the skin, continuously monitoring blood chemistry and transmitting data wirelessly to external devices. It's continuous diagnostics, real-time health monitoring, catching problems before they become emergencies, all because scientists figured out how to make human cells produce proteins that jellyfish use to glow in the ocean. Tissue engineering and regenerative medicine are using fluorescent proteins to track cell development. When scientists grow organs in the lab, and yes, that's actually happening now, not perfectly but increasingly successfully, they can tag different cell types with different colours. Stem cells glow one colour, differentiated cells another, and researchers can watch in real time as the stem cells transform, as tissues organise themselves, as structures that look increasingly like functional organs emerge from cellular precursors. It's like watching time-lapse footage of development, except it's happening in real time, and the colour coding helps distinguish cell populations that would otherwise look identical. Cancer researchers are using fluorescent proteins to study metastasis, how cancer spreads from primary tumours to distant sites. They tag cancer cells with red fluorescent protein, immune cells with green, blood vessels with blue, then watch the interactions in living animals. They can see cancer cells breaking away from tumours, entering blood vessels, travelling to new locations, and establishing secondary tumours. They can test drugs that might block metastasis, and watch whether the drugs actually work, whether they stop cancer cells from travelling, whether they make the red glow stay put instead of spreading through the body. You're back at the surface now, floating in calm water, watching the sun filter down through the waves above you. You're thinking about all the ways that bioluminescence, this weird evolutionary adaptation that lets organisms produce light, has been borrowed and repurposed by humans for applications that have nothing to do with why the light evolved in the first place. Jellyfish glow to defend themselves, to hunt, to communicate in the darkness. Humans took that glow and used it to see inside living cells, to guide surgical tools, to track diseases, to understand brains. It's technology transfer across kingdoms of life, taking solutions that evolution developed over millions of years and applying them to problems that evolution never encountered. And it works because biochemistry is universal, because the same molecular machinery that makes a jellyfish glow can make a human cell glow if you insert the right genes. Because life at the cellular level operates on principles that transcend species and habitat, and the original purpose of any given adaptation. There's a phenomenon called the deep scattering layer that's visible on ship sonar, a zone in the water column that reflects sound waves, creating what looks like a false bottom on depth sounders. For decades, sailors and researchers wondered what caused this layer. Turns out it's a massive aggregation of bioluminescent organisms, fish, squid, jellyfish, plankton, that migrate vertically through the water column every day. They rise toward the surface at night to feed, then descend to deeper, darker waters during daylight to avoid visual predators. This migration involves perhaps the largest movement of biomass on Earth, happening daily in oceans worldwide, and scientists can track it through bioluminescence patterns. Studying these migration patterns reveals information about predator-prey relationships, energy transfer through marine food webs, and how efficiently the ocean sequesters carbon. When organisms feed near the surface at night, they consume carbon-rich plankton. When they descend during the day and excrete waste or die in deep water, that carbon sinks toward the ocean floor, getting removed from the atmosphere for decades or centuries. It's called the biological pump, and it's a major component of Earth's carbon cycle. Bioluminescent organisms participating in this daily migration are thus directly involved in regulating atmospheric carbon dioxide, helping buffer climate change through processes that would be invisible without their glow marking their movements. Researchers have deployed underwater cameras in the deep ocean, stationary observation systems that sit on the seafloor for months, recording video of passing organisms. When they review the footage, they see bioluminescence everywhere, constant flashing and glowing from creatures moving through the water column. By analysing these patterns, how often organisms glow, what triggers the luminescence, which species produce which light signatures, scientists are discovering new species, documenting behaviours that would be impossible to observe any other way, and building databases of deep-sea biodiversity that reveal how much life exists in parts of the ocean we rarely visit. Some of these observations are startling. There are enormous gelatinous creatures in the deep ocean, siphonophores, jellyfish, and things that don't fit neatly into established categories, that only became known to science after deep-sea cameras captured their bioluminescence. These creatures are too fragile to survive being caught in nets, too transparent to see during submersible dives with normal lighting, but their bioluminescence gives them away, makes them visible against the darkness, lets researchers document their existence and begin studying their ecology. Climate change is affecting bioluminescence patterns in ways that scientists are just beginning to understand. Warming waters are shifting the ranges of bioluminescent species, bringing them into new areas, eliminating them from regions where they previously thrived. Changes in ocean chemistry, acidification from increased atmospheric carbon dioxide dissolving in seawater, may affect the calcium availability that some organisms need for their bioluminescent reactions. Researchers are monitoring these shifts, using bioluminescence as one more indicator of how rapidly ocean conditions are changing and what those changes might mean for marine ecosystems. There's also growing interest in using bioluminescent organisms as inspiration for environmental technologies. Imagine streetlights powered by bioluminescent bacteria instead of electricity. No power grid required, just nutrient-feeding systems keeping the bacteria alive and glowing. Or trees engineered with genes for bioluminescence, creating living lighting systems in parks and along roads. These applications are still theoretical, still facing technical challenges around brightness and maintenance and public acceptance, but they represent a direction where human technology might borrow not just the molecules, but the entire strategy of bioluminescence, producing light biologically rather than electrically, growing illumination rather than manufacturing it. The glow is steady, not flashing, because bacteria don't produce light in response to mechanical stimulation like dinoflagellates do. They glow continuously when their quorum sensing system is activated, producing a constant low-level luminescence that combines across vast populations to create the milky appearance. A single bacterium produces barely any light, far too faint for human eyes to detect. 1 million bacteria together might be just barely visible as a dim glow, but 1 trillion bacteria per cubic meter of surface water, spread across an area the size of Connecticut. That produces the Milky Sea effect, that turns the ocean into a glowing expanse that satellites can photograph from orbit. Why bacteria would maintain bioluminescence under these conditions is still debated. Some researchers think the glow attracts fish, and other organisms that consume the algae the bacteria are feeding on, creating mixing and circulation that helps distribute bacteria to new areas. Others suggest the light might serve signalling functions among bacterial populations, coordinating behaviour at scales we're only beginning to understand. Or possibly the glow is simply a metabolic byproduct that becomes visible when population densities reach extreme levels. Bacteria glowing not for any strategic reason, but just because that's what they do, and when enough of them are present, the cumulative effect becomes spectacular. Satellite monitoring is helping scientists understand how often Milky Seas occur, and where they tend to appear. Turns out they're more common than historical records suggested. Probably because many events happen in remote ocean regions where ships rarely travel, where there's nobody to witness the glow and report it. Researchers have identified dozens of potential Milky Sea events in satellite data from the past 2 decades, mostly in tropical and subtropical waters where conditions favour massive bacterial blooms. Climate change might be affecting Milky Sea frequency, though the data isn't conclusive yet. Warming waters and changing nutrient distributions could either increase or decrease the conditions that favour these bacterial blooms. Ocean acidification might impact bacterial metabolism and bioluminescence efficiency. Scientists are monitoring these patterns, adding Milky Seas to the list of oceanic phenomena that might shift in frequency or distribution as Earth's climate changes. You're still floating on your back, imagining what it would feel like to experience a Milky Sea directly. To be suspended above an ocean that glows beneath you with bacterial light, to see nothing but luminous water extending to every horizon, to drift through that eerie glow for hours while your brain tries to process something that looks impossible but is actually just biology happening at an extreme scale. It's one of the last great mysteries of bioluminescence, a phenomenon we're just beginning to understand through satellite technology and microbial ecology, a reminder that the ocean still contains surprises, still produces events so rare and strange that they seem almost mythological until science confirms them as real. You're back on land now, sitting somewhere comfortable, maybe your own room, maybe a quiet space where you can think about the future without interruption, and you're wondering what comes next. You've seen bioluminescence in jellyfish and bacteria, in fireflies and fungi, in the deep ocean and the Milky Seas. You've watched scientists borrow this light for medicine and research, but now you're asking the natural question, where does this go from here? What happens when humans stop just observing bioluminescence and start seriously engineering it, incorporating it into technology, building a world where biological light becomes as common as electrical light, where cities glow not from LED bulbs but from living systems producing their own illumination? This isn't distant speculation. It's already happening in laboratories around the world, just not yet at the scale that would make it visible in your daily life. Researchers have successfully inserted bioluminescent genes into plants. Tobacco plants initially because they're easy to work with genetically, but now ornamental plants, potentially trees. These plants glow faintly when you shine UV light on them, proving the genes work, proving the machinery is functional. The glow is still too dim for practical lighting. You couldn't read by a glowing plant, couldn't light a room with engineered trees, but the principle is established, the foundation is laid, and brightness is a problem that can be solved through further genetic engineering. Imagine walking down a street lined with trees that glow softly in the evening, not bright enough to feel artificial, just enough to provide ambient illumination that makes the sidewalk visible without streetlights, without power lines, without electricity. The trees would use photosynthesis during the day to generate the energy they need, then release it as light at night through engineered bioluminescent pathways. No power grid required, no energy costs beyond planting and maintaining the trees, no light pollution in the traditional sense because biological light can be tuned to wavelengths that don't disrupt wildlife or human sleep patterns, the way blue-heavy LED lights do. Scientists working on this have already identified the bottlenecks. Plant cells produce some of the molecules needed for bioluminescence, but not others. Luciferin needs to be synthesized through metabolic pathways that plants don't naturally possess, so researchers are inserting entire chains of genes, sometimes 5 or 6 different enzymes, to create the biochemical assembly line that produces luciferin from basic cellular building blocks. It's genetic engineering on a systems level, not just adding one gene, but adding entire metabolic pathways, turning plants into chemical factories that produce their own light-generating fuel. The brightness issue is also being addressed. Natural bioluminescence is dim because organisms don't need much light, just enough to communicate or startle predators or illuminate prey, but humans want brighter light. Light that competes with modern electrical systems, and that requires optimizing every step of the bioluminescent reaction. Researchers are engineering brighter luciferases, cells that produce more luciferin, systems that recycle the spent reaction products back into fresh fuel. They're borrowing from different organisms, taking the brightest luciferase from fireflies, the most efficient luciferin synthesis from bacteria, the best regulatory systems from jellyfish, and combining them in ways that nature never tried, creating hybrid bioluminescent systems that outperform any single natural example. Some companies are exploring bioluminescent bacteria as commercial lighting. Picture a lamp that's actually a sealed container of glowing bacteria, fed with simple nutrient solution, producing light continuously as long as the bacteria are healthy and population density remains high. The light would be soft, probably blue, green or white depending on which bacterial strains are used, and it would require no electricity, just periodic feeding with basic nutrients. It's a living lamp, a biological light source that grows rather than being manufactured, that can be composted rather than discarded as electronic waste when it reaches end of life. The challenges are significant, admittedly. Keeping bacteria alive and glowing consistently is harder than flipping a light switch. Biological systems are sensitive to temperature, humidity, contamination, pH changes, all factors that could dim or kill the glow. Brightness needs to increase substantially to compete with LED efficiency. And there's the practical question of whether people actually want living lamps in their homes, whether the convenience of electricity outweighs the novelty and environmental benefits of biological light. Consumer acceptance isn't guaranteed, even if the technology works perfectly. But imagine the applications beyond home lighting. Emergency lighting systems that don't require batteries or power, bioluminescent markers that glow continuously in tunnels, submarines, underground facilities where power failure could be catastrophic, self-illuminated exit signs that work during blackouts without backup generators, glowing crops that make nighttime harvesting possible without tractor-mounted floodlights, runways marked with bioluminescent strips instead of electrical lighting systems. Each of these applications leverages the fact that bioluminescence works without external power, works in conditions where electrical systems might fail, works continuously as long as the organism or the biochemical system remains functional. Synthetic biology companies are creating standardized bioluminescent parts, genetic constructs that can be inserted into different organisms to make them glow. It's like open source software but for biology, where researchers share genetic sequences and optimization techniques, where improvements get distributed across the scientific community, rather than being hoarded by individual labs. This collaborative approach is accelerating progress, turning bioluminescent engineering from experimental curiosity into practical technology, faster than would happen if every lab was working independently, reinventing the same solutions. There are artists working with bioluminescence too, creating living sculptures that glow, installations where viewers walk through spaces illuminated only by engineered organisms, performances where dancers wear clothing embedded with bioluminescent bacteria that leave glowing trails as they move. It's bio-art, living art, art that grows and changes and eventually dies, art that challenges assumptions about what materials artists can work with and what counts as a medium for creative expression. And then there's the more speculative stuff, the applications that sound like science fiction, but have serious research backing them. Bioluminescent crops that signal when they're stressed, changing colour to indicate drought or nutrient deficiency or pest infestation before visible damage occurs. Glowing roads that charge during daylight and illuminate themselves at night, reducing the need for streetlights. Trees engineered to glow near industrial sites, their light intensity responding to air pollution levels, creating a living monitoring system that makes environmental contamination visible to anyone who looks. Cities where buildings incorporate bioluminescent facades that produce ambient light without electricity, where parks glow softly at night from engineered plants, where the boundary between living systems and built environment becomes blurred. Some people find this exciting. This vision of a bioluminescent future where humans harness living light for practical purposes. Others find it unsettling, worried about releasing genetically modified organisms into the environment, concerned about unintended consequences, skeptical that biological systems can ever be as reliable as engineered electrical systems. Both reactions are reasonable. Bioluminescence technology is powerful, promising, and also requires careful consideration of safety, containment, ecological impact, and the social implications of blurring the line between natural and engineered life. You're getting very sleepy now, your thoughts drifting like bioluminescent plankton in a gentle current, and you're imagining a world where light comes from life, where the solutions evolution discovered over hundreds of millions of years become integrated into human civilization, where jellyfish glowing in the ocean lead somehow to trees glowing in cities, to crops that communicate through colour, to living systems producing illumination without consuming fossil fuels or rare earth minerals or contributing to the electrical grid's environmental footprint. It's a strange trajectory, from Luna pulsing in the deep ocean to defend herself from predators, to Equaria Victoria glowing green for reasons that probably involved sexual selection or prey attraction, to Shimamura squeezing thousands of jellyfish in Washington state, to GFP revolutionizing cell biology, to engineers trying to make trees bright enough to replace streetlights. It's technology borrowing from nature, except nature wasn't trying to create technology, it was just solving problems related to survival and reproduction. And humans are the ones seeing potential beyond the original purpose, seeing applications that evolution never anticipated or intended. And now, as you drift deeper into sleep, as the information settles and begins integrating itself into dreams that will probably feature glowing jellyfish and trees made of light, let yourself rest in the knowledge that bioluminescence is everywhere. In the ocean depths where Luna still pulses her blue-green signals, in laboratories where doctors use fluorescent proteins to see cancer, in forests where mushrooms glow faintly on decaying logs, in the future where engineers are building with living light, and in your mind where all these images are blurring together into something peaceful and strange and beautiful, like watching the ocean glow from a ship crossing milky seas, like floating through water where every movement creates sparkles, like the moment just before sleep when thoughts become less linear and more luminous, when consciousness fades into something deeper and darker and occasionally lit by flashes of understanding that pulse and fade like bioluminescence in the depths, like Luna's glow spreading through black water, like light produced without heat, without burning, just the cold, steady glow of life doing what it does best, adapting, surviving, and occasionally accidentally creating beauty in the process, creating light in the darkness, creating solutions that humans borrow and repurpose, and eventually build into technologies that might light cities, cure diseases, or simply help you understand that the natural world is stranger and more wonderful than most people realize, full of organisms producing light for their own reasons, while we watch and learn and wonder what else we might discover if we keep looking, keep asking why things glow, keep paying attention to the small bright creatures that most people never notice, but that contain within their cells the molecular machinery that might someday change how civilization produces illumination, how we understand biology, how we see the relationship between living things and the technologies we build from their borrowed parts. End of script. Sweet dreams.